Hi everybody, welcome to lab for March 24th. Today we're going to be going over the rest of the metabolic tests that we hadn't finished yet in class. So I'm going to walk you through those, show you what we're testing for, fill out our charts, and then show you what the results look like when we're finished. Before we get into today's metabolic tests, I just want to reiterate what the whole purpose of these is. So the goal behind metabolic tests is to identify bacteria. And we do this by using a lot of metabolic tests all together so that we can build a profile of a given bacteria. And so once we figure out all the test results, then we run them through a flow chart called a dichotomous key. And there's one of those shown here on the right. So then we can just follow the flow chart along. So we start out, for example, we might first look at the gram reaction. In other words, is our bacteria gram positive or negative? And then there's an arrow to follow for either results. So we might say, oh, we have a gram negative bacteria. And then we would go through a series of questions based on each metabolic test. So the first one might be glucose fermentation. And we would find out if we get acid or acid and gas. And then we might look at something like motility, or then we might look at something called the urease test, or is the urea hydrolyzed, or is indole produced, or is citrate utilized. All these terms you're seeing on here, urea hydrolysis, or indole use, or citrate, these are all metabolic tests. And so by doing a series of them, we can then plug that information into the dichotomous key, and eventually we'll get to the answer of what our bacteria is. So you can see that in these green boxes there's names of bacteria. So that tells us that if we start at the top and follow the results down for what we got, we'll end up with a bacteria. This is what we would have done had we been doing our unknowns. If we were having lab in person, you would be getting a mystery bacteria and then running a bunch of tests on it, building a dichotomous key, and by the end figuring out what your bacteria is. So I just want to run through a to-do list for you so you know what you're supposed to do in this virtual lab. So first, you should go through the pages in the lab manual and make sure you read over the introductions, get a general idea of what we do in the test, and then fill out the metabolic charts on pages 78, 89, 90, and 93. I will be putting pictures up of the metabolic charts, but just as we were doing in person, the key information you need to know about the tests is the information that you'll be plugging into the metabolic test, into the charts, and also what the tests look like. And I'll be showing you pictures of the test results so you can see them and learn to study them. I'm going to be holding you responsible for recognizing pictures of what the tests look like and knowing the information in the charts and the information on this PowerPoint. So for the first test we're going to look at is a test called the Methyl-Red-Voges-Proskauer test, or MRVP. This is a two-way test, which means that we start out with one bacterial culture and we actually run two tests on it. The things we're testing for are fermentation enzymes, which can take glucose and run them through a fermentation metabolic pathway. There are two possible pathways for glucose fermentation depending on what species of bacteria we're looking at. And those are shown on this chart here. So on the left, we start out with glucose. The first step in fermentation is glycolysis, and that gives us pyruvate. So all bacteria that do fermentation go through that first step. The key with the fermentation, as you might recall from class, is what happens to the pyruvate next. And there's a lot of different things that can happen to pyruvate. In all of them, we have NADH putting its electrons onto pyruvate, and then pyruvate rearranges its bonds. So really what this gets down to is how does pyruvate rearrange its bonds to handle the extra electrons that were dumped back onto it from NADH. So one of the pathways that's very common is something called the mixed acid fermentation pathway. And that leads to a lot of various acids that might end up in the growth medium. So possibilities for those acids include lactic acid and acetic acid, but there's others as well. The end result of this pathway is that the growth medium or the environment that the bacteria are in ends up quite acidic. The other pathway that's common is something called the 2,3-butane-diol pathway, and that ends up making a compound called acetoin. So with the MRVP test, we're using the MR part of the test to see if there are these mini acids, or in other words, did it go through the mixed acid fermentation pathway? And we use the VP part of the test to see if acetoin is produced. So if you're filling in your chart, which you ought to be doing, you should, on page 78, you can fill out this information. I've also put all of these charts on Canvas so you can look at them under the lab photos module as well. So again, just to summarize, the name of the test is the MRVP test. The thing that we're testing for, or the enzyme we're testing for, is the fermentation enzymes. And those can tell us, or the enzymes determine whether the bacteria does the mixed acid pathway or the 2,3-butane-diol pathway. So the broth that we're using is called MRVP broth. So we would inoculate our bacteria into that broth on day 1. After incubating the bacteria, then we would split that culture into 2 different test tubes. And in one of those test tubes, we would do the MR test. The MR test, again, tests for mixed acid. We add in a methyl red indicator, which tests for pH. It turns red at acidic pH. In the other tube, we would add reagents called alpha naphthol and potassium hydroxide. Potassium hydroxide is abbreviated KOH. That's the chemical formula for it. And then a positive test for MR would look like a color change from the yellow of the medium to red. And so that if you see a red color change on the MR test, that means you're positive for the mixed acid pathway. For the VP test, you will also look for a color change after adding the alpha naphthol and the potassium hydroxide. And that one is a color change to pinkish red, and that's positive for the VP test. A negative test for VP would be a kind of a brownish copper color. If you get a positive VP test, that means that the bacteria is using the 2,3-butane-diol pathway and making that acetoin as a result. So again, just to summarize again, on day 1, we start by inoculating our bacteria into this MRVP broth. And then after letting the bacteria grow, we split that broth into 2 tubes. So we take a milliliter and put it into one clean tube, and take another milliliter and add it to another clean tube. And then to each of these day 2 tubes, we would add the reagents for each test. So for the MR test, we add the methyl red indicator and flick it to mix it. And you should see a result immediately. In the other tube, the VP test, we add 25 drops alpha naphthol and 8 drops potassium hydroxide. You don't need to know exactly the number of drops, but you should know that the reagents that go into the VP test are alpha naphthol and potassium hydroxide. Normally for this test, we would shake it to integrate some oxygen and let it sit for 5 minutes. The color change develops over time, so you might have to go through several cycles of shaking and letting it sit before you see a color change. Eventually, though, both tests will be finished. So the MR test looks like this one on the left. So over here on the left, you see a positive test. So you can see this color change went from the yellowish of the medium to red. So that means this is positive for the MR test. On the right shows a negative test, and it's yellow. Okay, so and then the VP test looks like this area over here on the right, so I'll just zoom in there. So this tube on the left shows a color change to pinkish red. That's positive for the VP test. And then the color's a little hard to see here, but more of a brownish copper color is negative for the VP test. All right, so the next test we're going to look at is a test that looks at what happens when a cell breaks down proteins. And the first series of tests that address protein catabolism is something called it's a 3-in-1 test, and it's called the H2S indole motility test. So it's testing for 3 things. The first test, the H2S test, is testing for an enzyme called cysteine desulfurase. And this is an enzyme that converts the amino acid cysteine into pyruvate and hydrogen sulfide gas. The next test is the indole test. And this tests for an enzyme called tryptophanase. And the job of tryptophanase is to take the amino acid tryptophan and break that down into pyruvate and a compound called indole. Indole is a gas. And the third test is the motility test, and that tests for motility. We've talked about motility before. It's the ability of a bacteria to swim. This tests for motility in a tube rather than using a hanging drop slide, which is what we've used before. So because these 3 tests are done in one tube, the medium we use for this is called SIM medium. And SIM, it starts out yellow, and it refers to sulfide, which is derived from the hydrogen sulfide gas of this first test, indole, which comes from the second test, and motility, which is the third test. So SIM medium does these 3 tests together. The way these tests are set up is by getting a little bit of bacteria on your inoculating needle. And this, you haven't used the inoculating needle yet, but that's basically it's just the handle with a little wire on it. It doesn't have a loop on the end. So you would put a little bit of that on your needle, and then this is a solid medium. It's not broth, so this has agar. And you would stick that needle down into the growth medium about 2/3 of the way down and pull it back out. And then you would place a little strip of paper, and you can kind of see that on the edge of this test tube. That paper is coated in a compound called oxalic acid, and that oxalic acid is used to detect the indole in the second test. This slide here just shows ways of inoculating. So for the SIM test, we do what's called a stab. So that's when we use just our straight needle and just stab it down into the agar and pull it back out. Most of the other metabolic tests, we use a streak, which is where you take an agar slant. So this is agar that's solidified so that there's a large surface area on the top, and you would just do an S-curve back and forth to inoculate that way. Normally, you would use your inoculating loop for the streak. So the results of the SIM test that you're looking for depend on the test. So for the H2S portion of the test, you look for a color change in the growth medium from yellow to black. And it turns black because iron salts that are in the SIM medium react with hydrogen sulfide gas to produce a compound called ferrous sulfide. And that is a black precipitate. Actually, I'm going to show you a picture of that. So the positive H2S test is shown here on the left. So you can see this is originally on the right what it looked like. That's yellow. A color change is pretty obvious, a color change to black. So this tube on the left, the black one, would be a positive test for the H2S test, and the tubes on the right would be negative for the H2S test. For the indole part of the test, that's where this little paper strip comes in that you stuck over the edge of the tube. And as I said, that has oxalic acid on it. And if there's indole produced, then you should see a color change from white to pink on this strip. So that would look like this area over here on the right. So these paper strips here, the one on the left has turned pink, so that means it's positive for the indole part of the test, and the one on the right is white, which means it's negative for the indole part of the test. And then finally, for the motility portion of the test, you're looking in the tube where you've stabbed the culture, and what you're looking for is evidence that the bacteria have swum away from the place where you stabbed it. And when they swim away, they make the agar look kind of fuzzy, because you actually have the bacteria, you can see them having spread out or dispersed in the tube a little bit. So just to look at that a little more closely, if you look at these two tubes that are still yellow, the one on the right is negative for motility, and you can see this central line that's fairly distinct. That means that all the bacteria that were stabbed into the culture have stayed put. They have not started to swim away. So this tube on the right would be negative for motility, whereas this tube on the left, you can see cloudiness and fuzziness emanating from where you stabbed it. And that means the bacteria started here in the center, but then they've started to swim away. So it starts, as the bacteria move away, they cause cloudiness in the growth medium. So the fuzzy tube is positive for motility. The non-fuzzy tube is negative for motility. So you can look over this table to summarize what we've talked about. So again, for the hydrogen sulfide test, we're looking for the enzyme cysteine desulfurase. The product of cysteine desulfurase includes hydrogen sulfide gas. The growth medium is the SIM tube, and it's going to be the SIM tube for all three of these tests. The SIM tube contains iron salts that combine with hydrogen sulfide to form a black precipitate. So the positive phenotype for this test is a color change to black because of the production of hydrogen sulfide. For the indole test, we're looking for the activity of the tryptophanase enzyme that breaks down tryptophan into pyruvic acid and indole. To test this, we're using the paper strip that's coated with oxalic acid. And I've tried to draw up here what the paper strip looks like when it's bent over the edge of the tube. So it's just hanging above the agar. And a color change on the paper strip from white to pink indicates that the indole test is positive. And I would just indicate that the pink might be very light. So even if it's very light, it still counts as a positive test. And then for motility, we're testing really whether the bacteria have flagella or not. And a positive test on that would be a fuzzy line that's filling the tube, whereas the non-motile would have a non-fuzzy appearance, and the edge of where the stab is would be very distinct. So the next group of tests we're looking at is the phenylalanine deaminase test. This is also testing for the breakdown of an amino acid. In this case, it's the amino acid phenylalanine. And phenylalanine gets broken down by an enzyme called phenylalanine deaminase into a product called phenylpyruvic acid and ammonia. So for this test, we'll be streaking the bacteria onto the surface of an agar slant, letting it grow, and then the next day we add a reagent called ferric chloride, and we just drip the ferric chloride onto the growth at the surface of the slant. So when we do that, if the ferric chloride causes a color change from yellow to green, we say that it's positive for phenylalanine deaminase. If the growth medium stays yellow, then we say it's negative. So that's a pretty straightforward test, and you get the results immediately. Yet another test that looks at the breakdown of protein is the urease test. Urease is an enzyme that helps convert this compound called urea into ammonia and carbon dioxide. This is a fairly straightforward test as well. So you just use an agar called urea agar. It starts out kind of an orangish yellow color, and you streak the bacteria in an S-shaped line onto the surface of the slant. And if the urease enzyme is active, that means that you have a lot of ammonia being produced. And the ammonia is very alkaline, and there is a pH indicator in this growth medium that when it turns alkaline, it turns this bright pink color. If urease is not active, then that means the bacteria are producing acids instead of breaking down urea. So a positive urease test is this neon pink color, and a negative test is this yellow color. It is also possible sometimes with this test to get an orange-yellow result. That's considered negative for the purposes of this class. That happens when the urease enzyme is very slow acting. But for the purposes of our class, we're going to call the pink positive and the yellow negative. So those two tests are depicted on this chart here. We have phenylalanine deaminase. We use the phenylalanine agar, and then for the second day we add ferric chloride to the growth at the surface. For the positive test, it would turn from clear to green, and that means that's because the phenylpyruvic acid that's produced by the phenylalanine deaminase protein or enzyme is combining with this ferric chloride, and that would turn it green. For the urease test, we use urea agar, and we see a color change from orangish yellow to pink. That's because alkaline products are produced, specifically ammonia. The negative test would be orange, yellow, or yellow. Let me back up. This middle test, the rapid ornithine decarboxylase test, we are not doing, so you can just skip that. The last test we're going to talk about is the citrate test. This tests for a bacteria's ability to break down citrate using an enzyme called citrate permease. Specifically, that helps convert trisodium citrate, which is basically citric acid, into pyruvate. So for this test, we use an agar called citrate agar. It starts out green, and we streak the bacteria onto the surface using the inoculating needle that we bend into a hook shape. That basically makes it like a half loop, and we do that because it makes it so we don't put too much bacteria onto the surface. Because if you get too much onto the surface for this test, it can give you inaccurate results. So a positive test for citrate would be a color change from green to blue. So at these tubes over here, the one on the left is green. That's the negative test. And the one on the right is blue. That's the positive test. So we get a positive test because there is a production of alkaline products, including sodium carbonate and ammonia. So this growth medium actually contains a pH indicator that is turning blue when alkaline products are produced. One thing that can happen in this test is that, as I said, if there's too much bacteria, it's possible to get a partial change to a blue color. So that's why we do this half loop inoculation with the needle. So if you look at the chart for this test, the enzyme is called citrate permease, the agar is called citrate agar, and the positive test is a color change from green to blue, which again is produced through alkaline products, which are sodium carbonate and ammonia. And then just a note to know that if you get a partial change to blue, that could be that you got a false positive. Okay, so that's the end of our metabolic tests. Again, I would just point you to the to-do list at the beginning of this presentation. Make sure you can recognize what a positive test looks like based on these pictures that I've shown you, and then also fill in those charts and really use those as a study guide, because that will summarize the names of the tests and the enzymes, the reagents, and what the positive test looked like. Thanks for your attention.